So it's the Friday before spring break, and I have decided to just bust out of the mold a little bit, like pun intended. And rather than starting with any announcements today, I just want to show you some microbial art. And maybe the coolest of the microbial art is busting out of the mold, or is mold, in fact. We see a lot of these plates that are absolutely gorgeous. Maybe I should make it extra credit if anybody does some rad microbial art like this in micro, because we actually do work with some very beautiful bacterial strains. I actually have some cool photos, one of a Micrococcus luteus culture on a blood agar plate. So these are just beautiful images and an awesome place to begin our coverage of complex gene regulation. So remember when we talked last time, we were talking about how expression of genes is not something that a cell wants to do unless it wants to devote the energy to that process. You know, very much like Vibrio fischeri, when we talked about the fact that it only wants to express its lux operon when it is in an environment where lighting up gives it some sort of benefit. And certainly when it's in that symbiotic relationship with the squid, we recognize it as having a very big benefit for that energy input. So let's talk more about this complex gene regulation with picking up our coverage where we left off last time and talking about the way in which not only can repressors be allosterically regulated, but so too can activators. So activators can be modulated by either a positive regulation or a down regulation as well. Here I'm showing a situation where a positive induction of an activator is taking place. That is with the case of an activator, when a molecule, an effector molecule, binds the activator and causes it to bind the activator binding site, this enhances expression of the downstream genes. So we might say, to a certain extent, the opposite impact on an activator is felt by an inducer. That is, when an inducer binds to an activator, it enables binding to the DNA. However, when an inducer binds to a repressor, it keeps that repressor from binding to the DNA. In both cases, both of those impacts lead to induction, because it's only when an activator is bound and when a repressor isn't bound that we get full expression of downstream genes. That being said, we now recognize that one way to control gene expression is by changing the promoter sequence. The second way is by having DNA binding proteins that either enhance or repress the expression of downstream genes. So we're starting to count up these methods that affect gene expression or that regulate gene expression. And I'm going to bring up just jotted down document that shows this overview of mechanisms to control gene expression. And certainly these are the first two that I have listed. But they're not the only methods by which gene expression can be regulated. And another is a very nuanced way, and this is by production of specialized sigma factors. And in a sense, the specialized promoters that are recognized by these specialized sigma factors. Let me give you an example of what I mean by this. Remember that most promoters are recognized, the most usual promoter is that of TT-GACA and TADA-AT. However, it turns out that not every promoter looks like that, and in fact, not every promoter is recognized by the same sigma factor. So whereas the typical sigma factor, it's actually called sigma 70, has a real affinity for TT-GACA and TADA-AT, right? I mean, that is like its attraction to the max is to TT-GACA and TADA-AT. But there are other sigma factors besides sigma 70 that recognize very unusual promoter sequences. Let me give you one example, and I'm actually going to write it out. This is a heat shock promoter, or known as HSP for short. And at the -35 sequence of a heat shock promoter, the sequence, I don't have it memorized, so I'm looking here, is C, T, T, G, A, A. And you can see why I don't have it memorized, because it's nothing like TT-GACA really. And then at -10, the sequence that we see there is C, 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 A, T, N, T, A. Wait a minute, you're like, what the heck is N? N is a variable, so it could be, it could vary amongst a number of different nucleotides in that position. But yet again, this really looks nothing like a Tata box. So this particular unique sigma factor, or this particular unique promoter, is recognized by a particular unique sigma factor. So this is a heat shock promoter. And heat shock genes are only ever expressed under very rare circumstances. So the way in which a cell regulates or minimizes expression of the heat shock genes is to have a very unique promoter that is only recognized under unique circumstances by a very unusual sigma factor. So another way to control gene expression is with these specialized sigma factors. And the last one that I'm going to add in here that wasn't on the list a few years ago is this idea of accessibility. That is the epigenetics kind of hypothesis, or maybe even more than a hypothesis at this point, is the idea that genes, when much more tightly wound, are not as accessible for expression, and that winding and that packaging can be very much changed by environment. So this is our overview of ways to control gene expression. So writing this down then, certain sigma factors are only made under certain environmental conditions that then recognize very unique promoter sequences. These are often those genes that are involved in very unusual processes, such as response to extreme heat or a need to form endospores. So under that very going gets tough, tough get going kind of circumstances is when we see specialized sigma factors really playing a large role in regulation of gene expression. So without further ado, I want to jump into the idea of complex gene regulation. That is to think about an operon on which not only is there an activator binding site responding to the presence of an activator, but there is also downstream of the promoter an operator, and that operator responds to the presence of a repressor or the absence of a repressor. So let's talk about situations in which that complex gene regulation is very pertinent. So first off, catabolite repression is one of those circumstances. When we think about the term catabolite, essentially what that means is food source. That is something that can be catabolized or stripped and ripped of its high energy electrons. So what we think about that is catabolite repression. That is to say, when there is a food source that can be ripped and stripped of its electrons, it actually serves to repress gene expression. So one good example of that is when E. coli preferentially eats glucose, and therefore, in the presence of glucose, expression of its lactose degrading enzymes are repressed. So let's look at this in action. There's actually quite a great analogy for what occurs in E. coli cells when they're living in an environment where there is their preferred glucose source of energy, but there's also a secondary source of energy in the form of lactose, which it likes, but it's sort of its second favorite. It's not going to use it first or preferentially. So when E. coli is living in the presence of glucose, it's going to live on that glucose first. It's then going to pause, and then it's going to live off of lactose. So we get this pattern that we call dioxis growth, where E. coli is preferentially eating glucose, pausing, and then being like, okay, I guess I'll go ahead and head on to the lactose. This is very similar to when I take my ski team to a buffet, right? Because they're racing a lot and they're burning a ton of calories, sometimes as many as 4,000 or maybe even 6,000 calories per day. So you take them to a buffet, right? And when they go through this buffet line, the first time through the buffet line, they're going to eat what they preferentially like. then, they're going to pause, they're going to kick back for a minute, and then they're going to go back to the buffet, and they will have consumed what they preferentially like. They will then go on to what they don't like as well, and they'll consume that too because they're still hungry. And this is exactly what is true of E. coli. But when we think about what must be happening here, both glucose and lactose are clearly present in this environment at the same time. But only glucose is eaten first and not lactose. So what that must mean is that the glucose is literally inhibiting the cell's ability to consume lactose. That is to say, catabolite repression is in action. So what must be happening then there is that glucose is somehow serving as maybe a repressor, or maybe it's keeping the activator from activating. One of those two things must be going on there. Let's relate this to our lab coverage. And just this week, you all have inoculated KIA tubes. KIA tubes are such a pertinent system to think about the lac operon, because in a KIA, recall that we had lactose in excess and we had glucose in short supply. So what happens with an organism like E. coli is, of course, that it eats that glucose first. When it does so, it releases acidic byproducts of that catabolism, and it turns the slant yellow. However, it quickly runs out of glucose within about 8 hours of incubation, and it moves on and begins to eat the lactose at that point. Because E. coli is capable of eating the lactose, it continues to make acidic byproducts, and the slant and the butt of this tube stay yellow. So what happens in the E. coli cell is literally that at first the lac operon is turned off. That is, the genes needed to degrade lactose are not made. But after all that glucose is gone, the genes needed to eat lactose are upregulated. Now, Shigella dysenteriae can't eat lactose. It doesn't have a lac operon, right? So it is incapable of going on and moving on to the lactose. And that's why the tube stays red over yellow, because Shigella is not degrading the lactose. But we're curious to see what's going on in E. coli, where clearly the glucose is repressing the lactose operon, or the expression of the genes needed to degrade the lactose sugar. So we want to see what's actually happening on that lac operon as E. coli grows for, say, 24 hours in a KIA tube. So first off, we should make note that catabolite repression is the repression of expression of the enzymes that degrade lactose by glucose. But what's weird about that is that if we recall back to the lac operon that we drew earlier, and we talked about the lac operon as being a good example of mechanism 2, where a repressor typically binds to the operator. So here is our operator with our repressor that typically binds to that operator. But when lactose, the derivative of lactose, is around, we just made it a little black guy. What we said would happen is that the conformation of the repressor would change with the binding of lactose, and it would induce expression, right? Because it would keep the repressor from repressing. So we had genes 1, 2, and 3, all of these genes involved with the transport in and degradation of lactose. So we have our promoter here and the operator downstream. So we know already that lactose is serving to induce repression and to keep that repressor from repressing. So we find ourselves in a little bit of a confusing spot, because if we know that glucose is a repressor of this whole process, that is, glucose is what is partly keeping the lactose the lactose from being degraded or the lac operon from being expressed, we find ourselves at a confusing point because we know that the operator is already controlled by the presence of lactose. So it must be that glucose is not exerting its regulation at the operator. And that is the crux of the matter, is that catabolite repression is not being felt at the operator, and instead it must be being exerted at the other regulatory site, the activator binding site. But hang on a minute, glucose is not an activator, right? Not at all. In fact, the presence of glucose is inhibiting the expression of the lac operon. So how could we mold our head around the idea that glucose is inhibiting the expression of the genes 1 through 3, somehow inhibiting it at the activator binding site? Well, if we think about it, this is because it must be stopping the activator, I'll just call it A, it must somehow be stopping that activator from binding to the activator binding site. That is, the presence of glucose must keep the activator from activating, and that's how it inhibits the expression of genes 1, 2, and 3. So this is a bit confusing because if glucose is acting to stop the binding of the activator, it must not be what is being sensed directly. So glucose is not sensed itself. Instead, it uses a hitman, so to speak, a secondary messenger called cyclic AMP. And cyclic AMP and not glucose is sensed. And here's how this works. Cyclic AMP is present in the opposite level to glucose. So that is to say that they're inversely related. If glucose is high, cyclic AMP is low. If glucose is low, cyclic AMP is high. And so cyclic AMP serves to be the regulator. So if cyclic AMP is low, then we know glucose is high. And then we know that if glucose is high, the expression of the lac operon is repressed. Likewise, if cyclic AMP is high, then that must mean that glucose is low and that the lac operon is going to get upregulated at the activator binding site. So the enzymes needed to degrade lactose are synthesized when cyclic AMP is high. So now we start to recognize how this control might be exerted at the activator binding site. Here is a picture of the lac operon. And notice that the activator binding site at this current view is not bound by the activator protein. The activator protein is called Catabolite Activator Protein, or CAP for short. Catabolite activator protein has to be bound to the activator binding site in order to see full expression of the genes that degrade lactose. So that catabolite activator protein must in fact be regulated by the secondary messenger cyclic AMP. That is to say that if cyclic AMP is high, meaning glucose is low, then genes 1, 2, and 3 are going to be expressed because it makes sense to degrade lactose because glucose is low. So cyclic AMP is going to be the thing that allows the catabolite activator protein to bind. So the lac operon is under control of both an activator and a repressor. The activator, catabolite activator protein, is under control of the presence of glucose. So the CAP binding site is only filled by CAP when cyclic AMP binds to CAP and activates. Cyclic AMP being high means that glucose is low and that it makes sense to degrade the second preferred sugar, lactose, right? Move on to that on the second choice on the smorgasbord of foods. So not only is the lac operon controlled by the presence of lactose acting as an inducer to the repressor, but it is also controlled by the presence of glucose. And if glucose is there, CAP cannot bind. But if glucose is gone, cyclic AMP signals low glucose and it allows CAP to bind, saying, hey, let's go ahead and chow down on our second preferred source, lactose. So under conditions where lactose is absent, the repressor binds. Under conditions where glucose is absent, the activator binds. So in order to see full expression of genes 1, 2, and 3, we know that two things must be true. That is, first, lactose must be present, inducing the repressor, keeping it from binding. But we also know that glucose must be absent, allowing the cyclic AMP to be high and allowing CAP to bind to the activator. So in a KIA tube, when we first inoculate that tube, both lactose and glucose are present. So if lactose is present, the repressor is induced, that is, it is not bound to the operator. But if glucose is there, then we know that there is no cyclic AMP and CAP doesn't bind. Or said another way, we know that the cell would rather eat glucose, so it won't want to express the genes on the lac operon. So that is regulated by keeping the CAP from binding. Now, as the E. coli grows in the KIA tube, that glucose gets consumed and it is eventually used up. So once that glucose is used up, then the concentration of cyclic AMP is very high, and that enables CAP to bind. CAP binds, and we get the expression of genes 1 through 3. So in order to see full expression of genes 1 through 3, CAP has to be bound, that is, glucose has to be low, and the repressor cannot be bound, that is, lactose has to be present, keeping that from binding. So the lac operon gets fully transcribed only when both of those things are true. So at any given moment, there will be a certain basal level of expression of genes 1 through 3. That basal level is very, very low. In order to really see expression of genes 1 through 3, we have to see that both CAP is bound at the activator binding site and the operator has to not be bound by the repressor. That is, lactose has to be present, the derivative of lactose that binds there. I am fully aware that this is probably the cheesiest of the cheesy examples that I've ever used, but you know what? It's really late at night and I am like really a little bit loopy, and so I thought this would be a rad demo, so let's do it. So recognizing that the expression of the lac operon is dependent upon not just one, but two environmental conditions, we can make an interesting analogy. Okay, so let's make sure we understand that number one environmental condition that affects expression of lac operon is presence of lactose. Right? Genes 1, 2, and 3 are only going to be expressed in an environment where lactose is present. It doesn't make any sense to express genes to degrade a sugar that is not there. So the operator is the switch. You could think of it like a light switch. It is the switch that responds to the presence of lactose. If lactose is present, that switch is turned on. The other environmental condition that the lac operon responds to is the presence of glucose. This switch is at the activator binding site. And once again, genes 1, 2, and 3 are only going to be expressed when glucose is absent. That is, when cyclic AMP is present and the activator binds. So two circumstances have to be met in order to allow genes 1, 2, and 3 to be expressed. 1, lactose has to be present. 2, glucose has to be absent. So the analogy that I have here is a light analogy. This is one of my favorite demos from chemistry, showing that electrolytes can conduct a current. But the way that I'm using the example today is to show you that there are two switches that have to be on to make the light bulb light up. Number one switch will make an analogy that the presence of lactose is number one switch. That would be like me having plugged in this light. So I had to plug in this light in order for it to be able to light up. But notice that it's not lighting up. And that's because the presence of lactose alone is not enough to make the light light. That is, to make genes 1, 2, and 3, presence of lactose alone is not enough. However, if glucose is absent, making cyclic AMP high, then that is enough to make genes 1, 2, and 3 become expressed if both switches are on. So you can think about this glass of electrolytes, salt water, as being the absence of glucose, the presence of cyclic AMP. So unless the glucose is degraded and absent and lactose is present, we don't get the light. And that is a way that you can think about expression of the lac operon. Now let me make a quick caveat. Genes 1, 2, and 3 will always experience basal level expression. That is to say, a very small amount of expression will occur at any given time. But to get complete and total expression, both of those switches need to be on. Let me also qualify that gene 1 is what encodes for the enzyme to degrade lactose. It's called beta-lactamase. Gene 2 is what encodes for a permease to bring in the sugar. And gene 3 is a transacetylase whose function is not entirely well understood. But we do know that together those genes and expression of those genes are needed to utilize lactose as an energy source. So let's take a moment to look at a great example. We want to look at what the lac operon would look like if you grew this E. coli on a GSA, that is a glucose salt agar. Notice that glucose salt agar is pretty straightforward. It's glucose and salt, right? So glucose and salt is what we find in this medium. Glucose is the only sugar present. So what this means, I'm going to jot this down, is that glucose is present, lactose is absent. So wait a minute. When it comes back to our light analogy, if lactose is absent, it means this is actually unplugged, right? Completely unplugged. Glucose is present, that means light is not even in the salt water. So is this light by any means on? Absolutely not. Let's draw the operon showing what's happening there. So we have our 5' to 3' strand, our 3' to 5' strand. We have the activator binding site, the promoter, the operator, and genes 1, 2, and 3. So we recognize that neither switch is on. So said another way, glucose is present, meaning cyclic AMP is absent, meaning that CAP, or catabolite activator protein, is totally denied. Now at the operator site, remember that's responding to the presence of lactose. So activator binding site, that responds to the presence of glucose. Operator, that responds to the presence of lactose. But notice that the repressor binds to the operator unless a derivative of lactose is present, and lactose is absent, so the repressor binds. Switch here at the operator, off. Switch at the activator binding site, off. This is a double off operon. So what I want you to do for the last piece of your extra credit for this long week of vodcasts is I would like for you to add to your already evolving, already building extra credit email. I would like you to add on to that and tell me what the lac operon would look like if an E. coli cell were grown in a mannitol agar. That is to say that the only sugar is neither glucose nor lactose. So in this case, glucose is absent, lactose is absent, and I want you to explain to me what both the activator binding site, what's going on there, and what's going on the operator. That being said, I hope all of you have a kick-butt good spring break, and I hope I get some sleep pretty soon. So wait just a second before you all take off. I want to make sure that everybody knows that this is the end of where Exam 3 coverage stops, which gives you a lot of time to make a really great quality poster. So just a reminder that that poster can have all of the content that we've covered on it, and it can be of any style that you like. I'm going to actually show off one of my current teaching assistant's posters. She did a bang-up job on this poster, and what you're going to see is that she did it on the back of her lab coat. So this is an amazing example, colors included, that has all of metabolism up on the upper portion here. And then as you scan down, you're going to notice that she has really done it up with gene expression as well. So you've got all of your metabolism, including photosynthesis, and then at the bottom, the very bottom of the code, she's got all of gene expression. So this is a really rad example. Notice over here the lac operon of a really well put together poster on a lab coat. And by the way, now she wears it for all of her teaching. So those of you thinking about becoming a teaching assistant, which you should, will find this one appealing. Have a fantastic spring break one more time, and I will see all of you very soon.